The following is an AZPM original production.
For Arizona Public Media, I'm Tim Swindle, Professor Emeritus of Planetary Science at the University of Arizona, and this is Arizona Science. Joining me today is Bruce Tabashnik, the head of the university's Department of Entomology. Welcome, Bruce.
Oh, thanks, Tim.
Bruce, you have a history of trying to figure out how to combat pests that destroy crops. Most recently, you've been studying the corn earworm or cotton bollworm, an insect that attacks both corn and cotton. What has been the technique used to prevent the corn earworm from destroying corn crops?
One of the new techniques is genetically engineering the crop plants so that they make proteins that are normally made by bacteria. And those proteins are very specific in the way they kill insects. So they're environmentally friendly. My team studies how the pests evolve resistance to these genetically engineered crop plants because insects will adapt pretty much to anything we use to control them. So this is just another example of that Darwinian evolution, their ability to adapt readily to pretty much anything we throw at them.
How big of a problem is this pest?
This is one of the most economically damaging pests in the United States, and it causes hundreds of millions of dollars to damage to crops every year.
And what have you found about the corn earworm, about how it evades these proteins?
We know there are certain genes that have mutations that allow the insects to resist these bacterial proteins. These proteins have to bind in a very specific way to receptors in the gut of insects to kill them. So that's one of the reasons they're so safe. They also have to be ingested. So only insects that are actually eating the crop can even potentially be harmed by them. So the normal way that insects evolve resistance is they have mutations that affect those binding sites, those receptors. So it's kind of like a lock and key. It's like changing the lock, if you will, so the key, the toxin, can't fit and can't cause toxicity anymore. But that's not how the field populations of the corn earworm, or sometimes called cotton bollworm, like you said, that's not how they evolved resistance in the field to these bacterial proteins that the crops have been genetically engineered to make.
How do they evade it then?
We're not sure exactly. The most recent study we did, we used genomics, and we compared insects from the field that were either resistant or susceptible. We looked across the whole genome. And what we found is that there were 20 candidates, 20 genes that we know from other circumstances, either studying this very same insect in the lab or other insects that have evolved resistance to BT, either in the lab or field. And none of the 20, we call them the usual suspects, were guilty.
And do you have any idea what this portion of the genome does?
One hypothesis is that the trypsin genes, which there are 9 of them in a cluster, they're playing an important role. And this is a hypothesis because trypsin is a digestive enzyme that could either play a role in activating these proteins or degrading them. So if the degrading aspect was picked up, then that would be a way that they could help the insects resist the BT proteins. However, I have to say that we don't have good evidence for that. It's more a question of managing it more effectively. We want to manage it with minimal environmental harm, with minimal risk to human safety when we eat those crops, and also as efficiently as we can so growers can make a good profit.
Well, good luck with that.
Thanks. We might need it because this is one tough pest.
Our guest today has been Bruce Tabashnik, who's been studying ways to prevent crop loss to insect pests. This is Tim Swindle, and you have been listening to Arizona Science. You can also listen to this and other Arizona Science segments by going to the AZPM website at azpm.org.
AZPM's original productions are made possible in part by donations from listeners like you. Learn more at support.azpm.org. Thank you.